Aquatic ecosystems are essential for biodiversity, clean water, and climate regulation, and yet they are increasingly vulnerable to human activity. Within these systems, microbes are silent drivers of ecosystem health, especially in freshwater ponds. But how do we know when human impact is pushing them too far? As land use changes towards adding more roads, buildings, and managed landscapes, we as a species put increasing pressure on freshwater ecosystems. These changes can disrupt water chemistry, divide habitats, and stress organisms that upkeep these ecosystems. One group affected but often overlooked is the microbial communities, which are needed for nutrient cycling and ecosystem resilience. We've been using large-scale ecological monitoring to understand how landscape factors, including human land use, influence the microbial communities living in ponds. Sampling was done in multiple ponds across western PA, covering a variety of landscape types. Ponds are visited monthly during the summer, and water is collected and filtered to collect the microbes. I used CHIME2 to analyze microbial DNA from the pond water samples. Utilizing a barcode gene, I was able to create inventories of the species that live within these ponds. This was to look for patterns that correlate with environmental variables such as temperature, salinity, and road density. With that data, I used JUMP to generate plots for the data to quantify whether road density gathered from land satellite data and calculated through GQIS and other environmental factors demonstrated a noticeable correlation to microbe abundance in these ponds. We found a statistically significant relationship between road density and the microbial community structure, which species are present and how abundant they are, suggesting a direct human footprint on invisible biodiversity. While road density is the factor displaying this relationship, it is most likely a stand-in for human interference in these ecosystems. Next, we investigated the presence of microbial genes that allow for tolerance to heavy metals, with the prediction that these genes may be present in ponds influenced by human land use. I used PCR and gel electrophoresis to confirm the presence of heavy metal resistance genes in these samples. We observed the presence of this gene in some ponds, but not all, suggesting variation in microbial gene content across the landscape. Together, this work helps us identify ecological stress signals. By understanding how microbes respond to human changes, we can better protect water quality and interspecies health before it is too late. As land use continues to change, we need to pay closer attention to how even small organisms like microbes respond. Monitoring these shifts can help us catch early signs of ecological stress before it is too late to act.